The following is a selected video from masterthecontent.com, where you will find an extensive video library of lectures for a variety of standardized admission tests. We offer over 600 hours of detailed video lectures for a multitude of standardized tests. Use our interactive in-lecture table of contents to find specific topics of interest. Work through numerous in-lecture examples to help you internalize concepts. To learn more, visit masterthecontent.com. Your career, our passion.
And we are going to talk about RNA modification. So remember, I told you once the RNA transcript is formed in the case of eukaryotes, it's called pre-mRNA. It's immature mRNA. So it must undergo RNA processing. And now this processing begins as the RNA strand is still being transcribed. And for this processing to occur, or this processing consists of two main distinct steps. So the first step that occurs in the processing of mRNA, I'm talking about pre-mRNA, is called Rna capping. So what RNA capping is, it's basically the modification of the 5' end of the mRNA transcript by the addition of a guanine nucleotide to which a methyl group has been attached. So we call this structure the 5' cap. That is the 5' cap. So what happens is that a guanine nucleotide to which a methyl group has been attached is added to the 5' end of the mRNA transcript to form what we call the 5' cap. And that process is called RNA capping. That's the first thing that happens in the processing of mRNA. Now, the next thing that happens to process mRNA is called polyadenylation. Now, what polyadenylation does is that it involves sequence or two sequential enzymatic reactions. Now the first reaction will cut the RNA strand at a specific nucleotide sequence. So the first enzymatic reaction in polyadenylation will cut the RNA strand at a specific sequence. And the second enzymatic reaction, the second one, will add a series of repeating adenine nucleotides, about 50 to 250 of them, to that cut end to form what we call a poly-A tail. So to explain that, or to give you a better visualization of what I mean by RNA capping and polyadenylation, we have the diagram at the bottom of the screen right there. So this is the 5' end of what is a newly transcribed copy of RNA. This is pre-mRNA. So what happens in RNA capping is that a 5' cap, which basically consists of a nucleotide to which a methyl group has been attached, is added to this 5' end. So this is what we call the 5' cap. Now on the other hand, on the 3' end, what's going to happen is the process of polyadenylation. So two enzymatic reactions will occur. The first one will cut the mRNA transcript to end or complete the process of transcription, and the second enzymatic reaction will add this tail, the poly-A tail, the polyadenylation tail, to that 3' end of the transcript. And that polyadenylation tail usually consists of about 50 to 250 adenine nucleotides. So two things are happening. We're changing the structure of mRNA at both ends. We're adding a 5' cap on one end, the 5' end, and we're adding a poly-A tail at the 3' end. Now, these modifications don't happen for no reason. They obviously have a function. So we're processing mRNA because we need it to be in a different form before it can be utilized. So what these modifications are thought to do is, one, they're thought to increase the stability of the mRNA molecule. Remember, you're dealing with what can be a very volatile environment for macromolecules inside cells. So what these structural changes do is that they increase the stability of that mRNA molecule. They also assist or facilitate its exportation. Remember, mRNA in eukaryotes is produced in the nucleus, and it has to be exported through the nucleopores into the cytoplasm where it can be used. So making these changes, or adding the 5' cap and the poly-A tail, actually allows the nucleopore complex to identify mature mRNA and allow it to pass across the nucleopore into the cytoplasm, where it can then proceed to the process of translation. So export or exportation is also important, and that's another thing that RNA capping and addition of the poly-A tail helps to do. The last thing that it helps to do is also protect the mRNA. Remember, it's a volatile environment, like I said, so these ends will help protect the mRNA from degradation by hydrolytic enzymes, enzymes that degrade macromolecules such as mRNA. So they act as, or they perform a protective function. They also facilitate its export, and they also help in increasing its stability. Because remember, it's a very specific set of instructions. Each nucleotide triplet in mRNA dictates one amino acid. So you need that information, you need those instructions to be untampered with, you need them to remain in their initially formed state, and you need to make sure that nothing happens to distort that sequence. So these changes, such as RNA capping and polyadenylation, actually perform that function. So we're going to move on.